we are going to talk about the chemical properties of water, including acid-base properties and buffers. So you might remember from general chemistry that Bronsted-Lowry acids can donate protons. So here we have a Bronsted-Lowry acid donated a proton to another water molecule, which makes it a Bronsted-Lowry base because it is accepting those protons. So we call this autoionization. So water has both acidic and basic properties, with one water molecule acting as an acid, acting as a proton donor, and the other one acting as a base, or the proton acceptor. And what we get are hydronium ions and hydroxide ions. And we don't get that many of them, but they are measurable. So we can write this equilibrium, a constant expression right here, where we have the equilibrium of the two waters forming the hydronium ion and the hydroxide ion. And we get this equilibrium constant expression here, where we have the molar concentration of hydronium ions multiplied by the molar concentration of the hydroxide ions divided by the molar concentration of water squared. Now, the concentration of water is not going to change that much. So what we do is we fold in the concentration of the water and this equilibrium constant, and we give it a new special name called Kw, or K water. Biochemists tend to use H plus instead of H3O plus, but they are the same thing. The K water can be measured as 1.0 times 10 to the minus 14th at 25 degrees Celsius. So just as a side note, acid-base reactions are very rapid, and it's because protons are constantly being donated and being accepted by water molecules in solution. So they're sort of jumping from one water molecule to the other. So that means that they have great mobility, and that contributes to the observation that acid-base reactions are among the swiftest known. So just as a review here, if we look at the relationship between H plus ions and OH minus ions in solution, we know that when we multiply their concentration together, we get 1.0 times 10 to the minus 14th. That's a constant. So if the concentrations happen to be equal, then they will be both equal to 10 to the minus 7th molar. And we can express that as pH equals the negative log of 10 to the minus 7th molar. So a neutral solution has a pH of 7. If we have an acidic solution, that means that the proton concentration is greater than 10 to the minus 7th molar. And since their concentrations multiplied together need to be a constant, that means that the hydroxide ion concentration is less than 10 to the minus 7th molar, and acidic solutions have a pH less than 7. The opposite is true for basic solutions. So if the hydrogen ion concentration is less than 10 to the minus 7th molar, and then the hydroxide ion concentration is greater, the pH will be greater than 7. So we want to talk about acid dissociation constants, and we want to be able to define and describe strong acids and bases and weak acids and bases. And we also want to be able to discuss the pKa concept using acetic acid and ammonia as examples. So first, strong acids and bases. Strong acids and bases, I can give you a list. So hydrochloric, hydrobromic, hydroiodic acids, nitric acid, sulfuric acid, and perchloric acids are the strong acids. Strong bases are any hydroxides in the 1 or 2A group on the periodic table, except for beryllium hydroxide, which is weak. What they have in common is that they're virtually 100% dissociated in water. So that's what makes them strong. It has nothing to do with their concentration. It has to do with the fact that they are 100% dissociated in water. We can illustrate this using an ice table, where if I have 1 molar hydrochloric acid, really at equilibrium, I don't have much HCl intact at all. I really have 1 molar H3O plus and 1 molar Cl minus because of the stoichiometry of this reaction. If I wrote an equilibrium constant expression, I would essentially, though, be dividing by 0, which you can't do, or something very close to 0, which means the equilibrium constant, the KEQ value itself, would be very large. And it means it's not a very especially useful number. I will invite you to go to Top Hat to answer this question, but I'm going to go on with my recording. We can do the same thing with calcium hydroxide, or again, if I use my ice table, again, I don't have calcium hydroxide undissociated in the aqueous form anyway, in water, but I would have 1 molar calcium ion and 2 molar hydroxide ions. Again, if I were to write a Kb for this expression, I would be dividing by something 0 or very close to 0, and again, I get a value that's not especially useful. So again, I will invite you to go to Top Hat to answer the second question and the third question. But going on, what distinguishes weak acids and bases from strong? The weak acids and bases are not completely dissociated in water. In fact, they may be only very slightly dissociated. You may have a very large concentration of undissociated acid or undissociated base. So here the dissociation constants are useful because they describe the extent of the dissociation. How will you identify weak acids and bases? Well, if we're going to use the Bronsted-Lowry definition, a weak acid is a proton donor. So if you have a substance that can donate a proton, and it's not on the list that I gave you in that last slide, then it's a weak acid. Again, using the Bronsted-Lowry definition for bases, bases are proton acceptors. So in biochemistry, you want to look for something that has a lone pair of electrons, because if you're accepting a proton, the proton doesn't have any electrons and can't contribute any, so you'll need a lone pair of electrons that can contribute both electrons to the bond. So in biochemistry, look for nitrogens. They have lone pairs of electrons. So using acetic acid as an example, this is how you would have written the equation in general chemistry. The biochemists tend to write it like this, where you have acetic acid dissociated into H+ ions and acetate conjugate base. So here, if I write the equilibrium constant expression, and I call it Ka, and I multiply the molar concentration of H+ times the molar concentration of acetate, divided by the molar concentration of acetic acid, I can measure that, and it's 1.76 times 10 to the minus 5th. Here, the Ka is useful for describing the extent of the dissociation. But because they're small, we usually describe them in terms of pKa. So instead of saying that the Ka is 1.76 times 10 to the minus 5th, I can say the pKa is 4.75. So P is just a mathematical expression that means the negative log. So the pKa is the negative log of the Ka, and if you do the math, you get 4.75. So if I now look at a base, NH3, and look at its ability to accept a proton to become NH4+ or ammonium ions and hydroxide ions, again, I can write an equilibrium constant expression, and I can measure this in the lab, and I get 1.8 times 10 to the minus 5th. So again, here, this Kb describes the extent of the dissociation. However, biochemists tend to look at all species from an acidic point of view. So I'm more likely to talk about the acidity of NH4+ than the basicity of NH3. So if I look at NH4+ as an acid, as a proton donor, because acids are proton donors in water, if NH4+ donates a proton to water, we get hydronium ions and NH3. And I write my Ka, equilibrium constant expression, so the molar concentration of NH3 times the molar concentration of H3O+ divided by the molar concentration of NH4+. And I get 5.68 times 10 to the minus 10th. And I can calculate the pKa from the negative log, and I get 9.25. So you should note the Ka times Kb equals Kw, and just to convince you of that, I took the Kb from the previous slide and the Ka here, and if I cross out everything, I get K water. And if you do the math, the Ka times the Kb equals 1.0 times 10 to the minus 14th, which is the K water. So we can use the pKa values to compare the strengths of acetic acid and NH4+. And there is a top hat question on this, but I will tell you that acid strength refers to the ability to serve as a proton donor, because that's how we are defining acids, as proton donors. So if you look at these two, acetic acid is the stronger acid because it has a low pKa. We'll use the expression that we used in that article that I gave you, as we're going to define pKa as proton binding affinity. So a low pKa is low proton binding affinity. So if a substance has a low affinity for protons, it donates them readily. So this is how we're going to use pKa in this class. pKa is a measurement of the willingness of a species to donate protons. If a pKa is a low value, it has a low proton binding affinity. And if you have a low proton binding affinity, then protons are donated readily, and that will be a stronger acid. If you have a high pKa, that's a high proton binding affinity. So that means the protons aren't donated readily, but they're held. A biochemist would say that's a weaker acid, But you could also say that it's a stronger base, but we're going to use the acid point of view. So if pKa describes how readily protons are donated, and if pH describes the concentration of those protons, there must be a relationship between these two variables. And there is, and we can derive an equation called the Henderson-Hasselbalch equation. So I'm going to derive that now. So if I just draw the generic acid HA dissociates to form H plus and A minus, the Ka is equal to the molar concentration of H plus times the molar concentration of A minus divided by HA. And I can take that expression and I can take the log of both sides. So I have the log of Ka, and here I'm going to look at this as the product of H plus times A minus over HA. And mathematically, remember, if you do that, if you take the log of two numbers in a product, you would sum their logs. So this is the log of H plus the log of A minus over HA. The next thing I'm going to do is I'm going to take the negative, I'm just going to multiply everything by -1. So I get the negative log of the Ka, the negative log of the H plus, and the negative log of A minus over HA. Well, remember that negative log of Ka is pKa, and the negative log of H plus is pH. So I get this expression here. We can rearrange it to get rid of the negative sign, and we can say pH equals pKa plus the log of the molar concentration of A minus over the molar concentration of HA. But I need to make a slight adjustment because some acids like NH4 plus are positives and their conjugate bases are neutral. So I would really write this as HB plus donates a proton to form HB plus and B. So this form of the equation doesn't really work. So this is the form we're going to use. pH equals pKa plus the log of the concentration of the conjugate base divided by the concentration of the weak acid. So this is the Henderson-Hasselbalch equation. So you might remember from general chemistry that if you use ice tables, sometimes you had -x and +x terms that were ignored if they were too small. If they weren't too small, you had to use the quadratic formula to solve the equation. Here there are -x and +x terms here that I am ignoring. So they are small as long as the acid isn't too dilute and the pKa isn't too high. So we have some limitations, so we just need to recognize that. So in order to measure the pKa values experimentally, we need to do a titration. So the way we do this, you remember from general chemistry, is you take some acetic acid and put it in an Erlenmeyer flask, and then you take some standardized NaOH in your burette, and you would also put a pH meter probe in there. It's not shown in this diagram, but you would have to have that. And then you would carry out a reaction where you drip in the NaOH into the acetic acid, and then this reaction would take place where you have acetic acid and the base from the burette, and because this OH minus is strong, the reaction goes 100% to completion, so you get water and acetate. So as the titration progresses and we add more hydroxide ions, the acetate concentration will increase and the acetic acid concentration will decrease. We can describe any point along the way with the Henderson-Hasselbalch equation. And remember that pKa is a constant. So what we do is we take our data and we plot it against the volume of NaOH added. And we'll get something that looks like this. There's a link to the animated figure that I won't do here, but that's posted on Sakai. So if we start here with all acetic acid, as we add OH minus and we convert some of that acetic acid to acetate, we'll eventually get to a point where that's an inflection point on this curve, where we have the acetic acid concentration and the acetate concentration is equal. Past that, we're converting more and more of our acetic acid to acetate until we get to the end point in which we have all acetate. So that means we can use this equation. If we look at the midpoint, when these concentrations are equal, that ratio is 1. The log of 1 is 0. So this is how we determine the pKa. We just look at the halfway point and we just draw a line up and then draw a line over, and we can see that the pKa is a little less than 5. And if you remember from the slide I showed you before, the pKa for acetic acid is 4.75. So at the midpoint, the weak acid and the conjugate base are equal in concentration. So we can do the same thing for phosphoric acid. Phosphoric acid is a polyprotic acid, 3 protons. They don't all come off at once, they come off one at a time, and each one of them has a pKa value that describes the extent of the dissociation. So here, if we start out with all H3PO4 and add a full equivalent of base, we will pull off that first proton, but at the midpoint, again, draw a line up here and draw a line over, and you'll get the pKa of that first proton. Add a second full equivalent of base, and then you pull off the second proton, but again, at the halfway point, draw a line up and draw a line over, and you get the pKa of the second proton, and add a third equivalent and pull off that last proton, and again at the halfway point here you draw a line up and draw a line over and you get the pKa of that third proton. So this brings us to buffers, because you might have noticed that some of those pictures actually shaded in a buffering range. We define a buffer as a solution which resists changes in pH when small amounts of strong acid or base are added. The composition is either a weak acid and the salt of its conjugate base, or a weak base and the salt of its conjugate acid. So I'm going to illustrate both of those things here. So, but before I go on, I want to just illustrate buffer capacity, and that is just the definition of it. Refers to the ability of the buffer solution to resist changes in pH. So it's just related to concentration. So a 1 molar buffer has a greater buffer capacity than a 0.1 molar buffer, just because there are more buffering species. So if I have a weak acid, let's use acetic acid again, and the salt of its conjugate base would be sodium acetate, but the sodium is a counter ion, so we can disregard it. If I would add acid, H+, to my solution, the pH would change. Remember, pH equals the negative log of the H+ concentration. So the acetate effectively mops up the H+ to form more acetic acid. And it is true that I'm changing the ratio of the weak acid to the conjugate base to the weak acid ratio a little bit, and that's going to change the pH. But I'm not going to change it to the extent that I would if there were no buffering species present. If I were to add base to my buffer, then it would be the acetic acid's job to mop up the hydroxide ions, and here we get water and acetate. And again, we do change this ratio slightly. That will change the pH slightly. The pK, remember, is a constant, but we've mopped up the H+ and the OH-, and that would change the pH of the solution radically if those buffering species were not there. So to mop up both strong acid and strong base, the buffer has to consist of both a weak acid and a conjugate base. You need both of those species to be able to mop up hydroxide ions and hydrogen ions. Let me use another example of ammonia. And the salt of its conjugate acid would be ammonium chloride, but remember, chloride is a spectator, so we don't have to consider it. If we add acid to our solution, the acid is mopped up by the NH3 to form NH4+. If we added hydroxide, that is neutralized by the NH4+, which donates a proton to the OH- to form water, and we get NH3. And again, we're going to change that NH3, NH4+ ratio a little bit, but not nearly to the extent that we would if those buffering species weren't there. So again, we have to have both a weak base and the salt of its conjugate acid in order to mop up both the strong acid and the strong base. So we can ask the question, at what pH does a buffer work the best? It works the best when the weak acid and the conjugate base concentrations are equal, because then you have the greatest concentrations of both buffering species. And remember, we said that occurs at pH equals pKa. It also works fairly well on either side, plus or minus 1 of the pKa, because here you have a 1 to 10 or 10 to 1 ratio of the weak acid and the conjugate base. So, at pH equal pKa plus or minus 1, there are appreciable concentration of both. And remember, you do need both weak acid and conjugate base to mop up any strong base or strong acid, respectively. This graphic shows it fairly well. Here they're using X instead of A. But you can see at pKa plus or minus 1, we do have X minus, you have the conjugate base and the weak acid, HX. If we go too far below, we just have weak acid and we don't have any conjugate base. If we go too far above, we have conjugate base and we don't have weak acid. So you need to have both of these species. So at pHs much above or below the pKa, the buffer will not function effectively. So the pH of blood is held relatively constant by this carbonic acid equilibrium. So if you look at these species right here, hopefully you notice that you know that the pH of blood is 7.4, and you can see the pKa of this first association is 6.4. So this first reaction right here is going to be relevant, and the second one isn't. And if the pKa's don't convince you, hopefully this will. This is an acid-base distribution curve that shows the concentrations of all three of these species at pHs from 0 to 14. And if I draw a line up here at 7.4, you can see that I have some H2CO3 and some HC3O-, but I don't have any carbonate. If I were to extend this black line right here, you would see that the carbonate concentration at 7.4 is 0. So we don't have any carbonate concentration in blood. The blood buffering, the equation that is relevant for blood buffering is the dissociation of that first proton and not the second. So we have carbonic acid and we have bicarbonate, we have no carbonate. This is important because we can take that first dissociation and we can show that we can get carbonic acid by reacting water with CO2. That's a reaction that occurs in the absence of enzyme, but in the presence of carbonic anhydrase, it's a lot speedier. The other thing that we can note is this is tied to respiration because CO2 is relatively nonpolar and can cross through the membrane of the alveoli in the lung to the capillaries. So we have CO2 gaseous, which is gaseous CO2 in the lungs, is in equilibrium with CO2 aqueous, which is dissolved CO2 in the blood. In this dissociation of the three protons of phosphoric acid that we looked at, hopefully you can see that it's this second dissociation right here because the pKa is close to pH 7. So we will have H2PO4- and HPO4-2- buffering species. Here we don't have so much phosphate in blood, but we do have it inside cells. So these are the two phosphate species that are present in cells, not H3PO4 and not phosphate. So this is a nice graphic if you want to look at a lot of these acids and bases that we talked about today, and you can look at their pKa's, and you can look at pH 7, and you can see what kinds of things are relevant at relative pH, not just pH 7, but other pHs as well. So this shows you the relevant species at each of the pKa's of these acids and bases. So if you have a buffer in the cells that consists of the weak acid, H2PO4-, and its conjugate base, the HPO4-2-, this is the pKa2, which is 6.82. The way that this acts as a buffer is that if you add acid, the HPO4-2- mops up the acid to form H2PO4-. If you would add base, the H2PO4- dissociates a proton. It doesn't show that here, but this proton would react with OH- to form water. So again, in order to have an effective buffer, you need two species. You need a weak acid, H2PO4-, and you need a conjugate base, HPO4-2-, because each one of those species participates in the mopping up of H+ or OH-. So that's our lecture for today. So on Tuesday, we are going to do our first POGIL activity. It's in the yellow pages of your manual and also posted on Sakai. It's called acid-base distribution curves, like the one I just showed you. We will do key questions 1 through 5 for homework. So when we get to class, we will complete questions 6 through 19 in class on Tuesday. So I will see you then.